Let's once again learn about the cell membrane, chemical composition, molecular organization of plasma membrane, lipid bilayer model, trilaminar model of Danieli and Davison, Robertson's unit membrane concept, fluid mosaic model, and membrane proteins, along with a conclusion about all these topics. So first, cytoplasmic surface of all living cells is bound by a limiting membrane called plasma membrane. It is elastic, porous, and semi-permeable. It is of lipoprotein composition, semi-fluid consistency, laminar or micellar organization. Interface between exterior and interior of the cell and thereby regulates the exchange of materials between cell and its external medium. Plasma membrane present in all types of living cells. In blue-green algae, bacteria, fungi, and plants, a cell wall is present. In animal cells, cell wall is absent. So, cell membrane is a biological membrane that separates and protects the interior of cells from the outside environment. Cell membrane consists of a lipid bilayer made up of two layers of phospholipids with cholesterol interspersed between them. Membrane also contains membrane proteins, including integral proteins that span the membrane and serve as membrane transporters, and peripheral proteins that loosely attach to the outer side of the cell membrane. And we'll just learn about the chemical composition. It's formed of lipids, proteins, carbohydrates, and water. So there are membrane lipids. Lipids have a structural role. Permeability barrier includes phospholipids, glycolipids, sphingolipids, plasmalogens, sterols, etc. Next is the phospholipids. Membrane phospholipids are the derivatives of glycerol, which is a 3-carbon alcohol. So they are called phosphoglycerides. A phospholipid molecule is formed of a glycerol molecule, 2 fatty acid molecule, and a phosphorylated alcohol complex. Glycerol forms the backbone. Glycerol forms the backbone. Phosphate group complex forms the head of the phospholipid polar and hydrophilic molecule, and the fatty acid forms its tail, non-polar and hydrophobic region. Here we can see a diagrammatic representation of phospholipid, fatty acid, and saturated or unsaturated glycerol along with the phosphate. General structure of a phospholipid. A phospholipid has a similar structure to triglyceride but has only 2 fatty acids and a phosphate in addition. So when we learn about glycolipids, exclusively restricted to the non-cytoplasmic outer side of plasma membrane, gangliosides are perhaps the most complex type of glycolipids. Sphingolipids are there. Sphingolipids are similar to phospholipids in structure, and the only difference is that in them glycerol is replaced by sphingenin as a core molecule. Then there are sterols. Sterols are non-polar and absent in prokaryotes. Cholesterol is there, exclusively found in the animals and the most abundant animal sterol. It is widely distributed in all cells and is a major component of cell membrane and lipoprotein. Cholesterol is first isolated from bile. Cholesterol literally means solid alcohol from bile. Structure and occurrence: C27H48O as a structural component of plasma membranes. Cholesterol is an important determinant of plasma membrane permeability properties. It is a yellowish crystalline solid. It is insoluble in water and soluble in organic solvents. It's a poor conductor of heat and electricity. It's present in abundance in nervous tissues. It appears that cholesterol functions as an insulating cover for the transmission of electrical impulses in the nervous tissues. Has a role in the synthesis of bile acids, hormones, and vitamin D. Next, lipid membrane is there. Sugar residues and lipid residues are attached along with glycolipid. Then membrane protein. Membrane proteins are mostly globular without orderly arrangement. They are randomly dispersed and discontinuous. They remain partially or completely inside and outside the lipid bilayer. The proteins completely outside the lipid bilayer are called extrinsic or peripheral proteins. Those which penetrate the lipid bilayer is called integral proteins. Integral proteins amount to 70 to 80% of membrane proteins. Some integral proteins span the entire lipid bilayer. They are called transmembrane proteins. So they form pores through the membrane and also transmit signals from one surface of membrane to the other. Intrinsic proteins usually float and freely move about within a lipid matrix, just as icebergs float and drift in the open sea. The dispersed distribution of proteins forms the mosaic part of the fluid mosaic model. Extrinsic proteins are hydrophilic. They bind with the membrane surface by electrostatic interaction and hydrogen bonding. Most intrinsic proteins are amphipathic. So phospholipids are there, peripheral protein is there, channel protein is also there. So carrier protein is associated with that, integral proteins, and the glycoprotein is there, cholesterol is there. This is a diagrammatic representation. Phosphate head, polar hydrophilic portion, fatty acid tail is a non-polar hydrophobic portion. When we just diagrammatically represent this, draw phospholipids, peripheral protein, channel protein, carrier protein along with glycoprotein, along with the cholesterol molecule and integral proteins. Their hydrophobic portion resides within the lipid bilayer. Hydrophilic part projects out of the lipid bilayer. Lipid anchored proteins, also known as lipid linked proteins, are proteins located on the surface of the cell membrane that are covalently attached to lipids embedded within the cell membrane. Lipid anchored proteins, also known as lipid linked proteins, are proteins located on the surface of the cell membrane that are covalently attached to lipids embedded within the cell membrane. These proteins insert and assume a place in the bilayer structure of the membrane alongside the similar fatty acid tails. Lipid anchored proteins are there. Phospholipid bilayer can be seen, and a peripheral membrane protein is there. Then integral membrane protein is there. Peripheral membrane protein and lipid anchored protein along with phospholipid bilayer. Functions of membrane proteins: transport of nutrients, passage of water, selective transport of molecules, maintenance of proper ionic composition inside the cell, reception of signals from the extracellular environment, expression of cell identity, physical and functional connection with other cells or extracellular matrix in multicellular organisms. Molecular organization of plasma membrane. When we learn about the molecular organization of plasma membrane, we can see that it has a lamellar or micellar organization. Lamellar hypothesis holds that the membrane is formed of molecular laminations, that is sheet-like molecular layers or lipids and proteins. Micellar hypothesis holds that it is formed of globular subunits of lipoprotein. Several models have been proposed to explain the molecular organization of plasma membrane. Fall under two main categories, lamellar and micellar. So membrane models are of two types, lamellar models and micellar models. Lamellar in the sense a sheet-like molecular level of lipids and proteins. So lipid bilayer model and trilaminar sandwich models are there. This trilaminar sandwich models are of four types, like Danieli-Davison model, unit membrane model, greater membrane model, and lipid pillar model. And micellar models are of four types, like Benson's model, Lennard-Singer's model, mosaic membrane model, and fluid mosaic model. So first one is the lipid bilayer model of Gorter and Grendel. In 1926, Gorter and Grendel postulated the lipid bilayer hypothesis. According to it, cells are covered all around by a 2 molecules thick lipid layer. In this bilayer, the polar ends of the molecules are directed outward and the non-polar ends inward. So extracellular region is there, intracellular region is there. In between them is the phospholipid bilayer. Along with that, hydrophobic tail is situated inside and hydrophilic head is outside. Trilaminar model of Danieli and Davison, that is the sandwich model proposed by Danieli and Davison. According to this concept, plasma membrane is 3-layered structure. It consists of a central phospholipid bilayer set between 2 peripheral protein monolayers, just as butter or egg in a sandwich. The lipid molecules of one layer remain linked in those of the outer layer by their nonpolar tails. The lipid molecules of one layer remain linked with those of the other layer by their nonpolar tails to van der Waals forces. They link with the proteins by polar ends through ionic linkages, hydrogen bonds, or electrostatic forces. So globular protein, hydrophilic zone, hydrophobic zone, hydrophilic zone. Next, Danieli and Davison's model. There are electrostatic interactions between the polar end of each lipid molecule and charged amino acid side of the phosphopolypeptides of the surface proteins. Other groups could be bound to the outer protein surface by their electrostatic or van der Waals bonds. According to this model, lipid bilayer could be about 6 nm in thickness. Each of the protein layers of about 1 nm thickness gives a total thickness of about 8 mm. Then, in spite of this very thin membrane is an efficient barrier to the movement of certain substances. Such a membrane would be capable of distinguishing between molecules of different size and solubility properties, and also between ions of different charges. So the cell membrane exhibits selective permeability. Protein coat lipid bilayer is there, polar pore, Danieli-Davidson model is here. So when we just try to explain the Danieli-Davidson's model, evidence to support this model: when high magnification electron micrograph of membrane become available, it appears to show the evidence of this model. It shows three-layered structure where a lighter color region can be observed with two dark parallel lines. Proteins appear dark, whereas a lighter color region can be observed between two dark parallel lines, and phospholipids appear light, possibly indicating protein layers either side of the phospholipid core. So lighter layer, darker layer is there, second cell membrane, intracellular space, first cell membrane, electron micrograph of plasma membrane. Evidence to support this model: when high magnification electron micrograph of membranes become available, it appears to show the evidence of this model. Even the total thickness of the membranes turned out to be about 7.5 mm, which is very close to 8 mm proposed by this model. Next is the Robertson's unit membrane concept. David Robertson proposed this unit membrane concept. The model proposed by him is mostly based on the Danieli-Davidson trilaminar concept. Robertson suggested that all biological membranes have the same trilaminar structure with approximately 75 angstrom thickness. To such a universal membrane, we give the name unit membrane. Central zone and marginal zone are there. So central zone is a lipid bilayer described by Robertson as the lipid bilayer leaflet. Here is a picture associated with this topic. Extracellular medium is their outer protein layer, hydrophilic heads of lipids, hydrophobic tails of lipids, phospholipid bilayer, intercellular cytosol, inner protein layer. Unit membrane concept. The central lipid core of the membrane is structurally and functionally important. Proteins do not have significant structural and functional roles. Proteins and lipids have perfect symmetrical and continuous arrangement. Even though basic ideas of the model are still valid, their trilaminar model seems to be oversimplified scheme. In this, proteins are entirely outdyed lipid bilayer. Studies have shown that protein and lipids are asymmetrical in arrangement. Robertson's model or unit membrane model in the late 1950s, electron microscopy provided additional information about the structures of the plasma membrane. David Robertson proposed this famous unit membrane model after studying the cell membranes from electron micrographs of mentioned material. He concluded that the plasma membrane and the membranes of all cell organelles were similar in structure. According to him, the cell membrane is trilaminar, made of three layers like protein, lipid bilayer, and protein. The physical arrangement of this trilaminar model has led to the term unit membrane. So Robertson's model or unit membrane model, this transparent lipid bilayer is surrounded by either side by denser proteins with a definite difference in type of their outer and inner side. Each of these two external hydrophilic protein layers is of 2 nanometer length, and the polar groups of phospholipids of one layer of the bilayer is by about 0.5 nm. So total external hydrophilic layer is about 2.5 nm thick. The middle layer is a hydrophobic bimolecular layer made of phospholipids without the polar groups, is 2.5 nm to 3.5 nm in thickness. The total breadth of the cell membrane is 6.5 nm to 8.5 nm. Which are the shortcomings of this particular model? Unit membrane is an oversimplified version. It does not satisfactorily explain the permeability and transport properties of the membrane. It simply regards the membrane as a rigid and inert structure. It does not account for the dynamic nature of the membrane and also for the molecular mobility of its protein and lipid constituents. It simply considers the membrane as a static structure. And we learn about the fluid mosaic model. This is a very important one, proposed by Singer and Nichols in 1972. According to it, plasma membrane is a two-dimensional quasi-fluid solution of organized liquids. Two-dimensional quasi-fluid solution of organized lipids and globular proteins. Plasma membrane is a bimolecular quasi-fluid lipid structure in which proteins freely float, move about and interact. So which are the major features? Plasma membrane is a dynamic structure existing in a liquid, crystalline or quasi-fluid state with mosaic molecular arrangement and free molecular movement. So globular protein is there, glycoprotein is there, carbohydrates are there, glycolipid, cholesterol will be there, integral protein is there, peripheral protein, channel protein, alpha helix protein, etc. are given. Fluid mosaic model, when we just illustrate this with a figure. Extracellular fluid, carbohydrate, transmembrane protein is there, glycolipids are there, glycoprotein is present, peripheral protein is there, cholesterol is there. The fluidity of the membrane is directly proportional to the chain length and unsaturation of fatty acids. The major components of the membrane are held together through non-covalent interactions. The cohesive forces between lipids are also between lipids and protein are weak hydrophobic interactions like ionic and hydrogen bonds. Membrane lipids and proteins undergo free lateral diffusion. Vertical diffusion of proteins and lipids is very much restricted. Membrane phospholipids are arranged in a bilayer that serves as a structural framework, a permeability barrier, and also as solvent for integral membrane proteins. Membrane lipids interact with proteins for the stabilization and proper functioning of the membrane. According to the Singer-Nicolson concept, phospholipid molecules have a continuous arrangement. They are arranged side by side and back to back in parallel rows, forming a continuous lipid bilayer. This lipid bilayer forms the structural framework of the membrane. So, in its outer monolayer is formed mostly of acidic phospholipids, and the inner ones consist of natural phospholipids. Their polar heads are directed outward to the aqueous phase. Non-polar fatty acid tails are directed inward to form the non-aqueous central core. This arrangement enables maximum interaction between polar heads and water molecules. The two lipid monolayers remain linked with each other by their fatty acid tail through hydrophobic interaction forces. So extracellular fluid is there, carbohydrate, transmembrane protein, glycolipid, glycoprotein, along with peripheral proteins, cholesterol, filaments of cytoskeleton, and cytoskeleton itself can be seen. The membrane phospholipid layers are in a fluid mosaic mobile fluid state, free to diffuse laterally within the molecule. The phospholipid layers is essentially a film of oil and it makes the membrane semi-fluid. Cholesterol occurs tightly bound in phospholipids. And we just tried to learn the structure: protein channel, transporter protein, globular protein, glycoprotein, carbohydrate, hydrophilic heads, phospholipid bilayer, phospholipid molecule, hydrophobic tail, alpha helix protein or integral protein, surface protein, filaments of cytoskeleton, integral protein, peripheral protein, glycolipid, cholesterol, etc. Singer and Nicolson model, like fluid mosaic model. In 1972, Seymour, Jonathan Singer, and Nicolson published the fluid mosaic model of the structure of cell membrane. The fluid mosaic model expanded on the Danieli-Levinson's model by excluding transmembrane proteins and eliminating the flasking protein layers that were not well supported by experimental evidence. The model remains in the current accepted model of the plasma membrane. Here the arrangements of the phospholipids remain the same, but the location of proteins is different. The model shows that these assemblies are highly dynamic. That is, this arrangement is fluid, not solid. There are different types of molecules put together in cell membrane and known as mosaic. These molecules are constantly moving in two dimensions in a fluid fashion, similar to icebergs floating on the ocean. The movement of the mosaics of molecules make it impossible to form a completely impenetrable barrier. So Singer and Nicholson's fluid mosaic model. According to the model, phospholipids can rotate or diffuse laterally, which happens very rapidly or transversely. Flip-flopping, rotation, lateral diffusion, swinging movement, transverse diffusion, etc. happen. The PM is composed mainly of amphiphilic, having both of the parts hydrophilic and hydrophobic phospholipid molecules. Phospholipids are the main fabric of the membrane, primary major component. Like glycoprotein is there, carbohydrates, glycolipid, cholesterol, globular protein, alpha helix protein, channel protein, peripheral protein, or integral protein associated with phospholipid bilayer. The mosaic of a This mosaic of phospholipids, cholesterol, proteins, and carbohydrates that gives the membrane a fluid structure. Their properties varies with cell type. So, glycoprotein, carbohydrate, glycolipid, cholesterol, phospholipid bilayer, integral protein, peripheral protein, channel protein, alpha helix protein, and globular protein is there. Cholesterols are attached between phospholipids as well as between two phospholipid bilayers. Cholesterol is only found in animal cells. This model shows that proteins are embedded in the bilayer as integral, peripheral, and lipid-anchored proteins. Lipid-anchored proteins are located on the surface of the phospholipid bilayer and are covalently attached to lipid molecules embedded in the layer. Evidence to support fluid mosaic model. Phospholipid bilayer in membrane. The electron micrograph of plasma membrane show that there are two prominent dark lines which indicate the presence of phospholipid bilayer. Intercellular space, first cell membrane, second cell membrane. Along with that, phospholipid bilayer is there, lighter layer phospholipid tail and darker layer phospholipid heads. Phospholipid bilayer in membrane. In freeze fracture experiment also shows the evidence for phospholipid bilayer because the fraction occurs here along the lines of weakness along the middle of phospholipid layer. Then there are also mosaic-like structures. When biological membranes are freeze fractured, there are some bumps, grooves, ridges seen inside the fracture plane of membranes. It is a mosaic-like structure consisting of a smooth matrix but interrupted by a large number of particles. Membrane fluidity. Membrane fluidity can be demonstrated through two experiments. Fluorescence discovery after photo-bleaching, like FRAP. A small section of phospholipids is bleached and become distinguishable from the rest of the phospholipids. Over time, the bleached area will begin to disappear as the lipids begin to recover. Membrane fluidity. Membrane fluidity can be demonstrated through two experiments. Fluorescence recovery after photo-bleaching. FRAP, fluorescence recovery after photo-bleaching. This occurs because the lipids are naturally diffusing throughout the membrane, including the bleached area. Membrane fluidity can be demonstrated through two experiments like differential surface antigen labeling and cell fusion. In this experiment, cells are taken from two different organisms and thus have different detectable surface antigens. Then they are forced to fuse. Now one can detect that the antigens diffuse throughout the membrane. This indicates that the lipids are laterally diffusing through the membrane. Antigens are there, so unfused, hemifused, and fully fused. Cells are taken from two different organisms and thus have different detectable surface antigens. They can have forced diffuse. Now one of them can detect the antigen diffuse toward the membrane. This indicates that the lipids are laterally diffusing through the membrane. Next, functions. Compartmentalization. The plasma membrane encloses the contents of the entire cell, whereas the nuclear and cytoplasmic membranes enclose diverse intracellular spaces. The plasma membrane encloses the contents of the entire cell, whereas the nuclear and cytoplasmic membranes enclose diverse intracellular spaces. The plasma membrane encloses the contents of the entire cell, whereas the nuclear and cytoplasmic membranes enclose diverse intracellular spaces. The various membranes bound compartments of a cell possesses markedly different contents. Membrane compartmentalization allows specialized activities to proceed without external interference and enables cellular activities to be regulated independently of one another. Provides a selectively permeable barrier. Membranes prevent the unrestricted exchange of molecules from one side to the other. At the same time, membranes provide the means of communication between the compartments they separate. Transporting solutes. The plasma membrane contains the machinery for physically transporting substances from one side of the membrane to another, often from a region where the solute is present at low concentration into a region where the solute is present at much more higher concentration. The membrane's transport machinery allows a cell to accumulate substances such as sugars and amino acids that are necessary to fuel its metabolism and build its macromolecules. The plasma membrane is also able to transport specific ions, thereby establishing ionic gradients across itself. This capability is especially critical for nerve and muscle cells. The membrane's transport machinery allows cells to communicate substances such as sugars and amino acids. Specific ions are also transported. Responding to external stimuli. The plasma membrane plays a critical role in the response of a cell to external stimuli, a process known as signal transduction. Membranes possess receptors that combine with specific ligands or respond to other types of stimuli such as light or mechanical tension. Different types of cells have membranes with different receptors and are therefore capable of recognizing and responding to different environmental stimuli. The interaction of plasma membrane receptor with an external stimulus may cause the membrane to generate a signal that stimulate or inhibit internal activities. For example, signals generated at the plasma membrane may tell a cell to manufacture more glycogen, to prepare a cell division, to move forward a higher concentration of particular compound, or release calcium ions in internal stores, etc. Cellular interaction. Situated at the outer edge of every living cell, the plasma membrane of multicellular organisms mediates the interaction between the cell and its neighbors. The plasma membrane allows cells to recognize and signal one another, to adhere when appropriate, and to exchange materials and information. Energy transduction. Membranes are intimately involved in the process by which one type of energy is converted to another type, energy transduction. The most fundamental energy transduction occurs during photosynthesis, when energy in sunlight is absorbed by membrane-bound pigments, converted into chemical energy, and stored in carbohydrates. In eukaryotes, the machinery for this energy conversion is contained within membranes of chloroplast and mitochondria. So, which are the functions? Energy transduction, intercellular integration, responding to external stimuli, transporting solutes, providing a selectively permeable barrier, etc. Providing a selectively permeable barrier, transporting solutes, responding to external stimuli, intercellular interaction, energy transduction.